what you're going to get now is not a lot of data, but you're going to get some conceptual sort of ideas and understanding, hopefully, of what goes on below ground and how that contributes to soil health. So with that, we'll just get going. Soil health as a concept or a terminology is relatively new in the US, but it actually has been around in Canada and Europe for a lot longer. We chose early on to go with the soil quality term, and that was sort of in the forefront of this type of science to begin with in the 90s and 80s. If you look at this little diagram here, a lot of the earlier work on soil quality was really more of a visual or a tactile assessment. Then when we started to coin more of the soil quality terminology and started to think about indicators and how we could follow things through time and space, we began to include more chemistry and biochemistry and maybe some physics, physical properties. It really wasn't until around 2010 and maybe a little later that the terminology sort of got shifted towards this idea of soil health. And that was really paralleling what was going on in the medical fields, which has been a real boon, I think, to understand more holistically the health of human beings and animals and so on and so forth. So we've been trying to do that in the realm of soils. And it's been great for us soil scientists to see our object of study getting so much recognition in a broader context. And the one other thing that was quite different is that we really started to appreciate the biology. Myself, I'm a fully trained soil scientist in all aspects of soil science and spent a lot of time doing soil chemistry and soil organic matter in my earlier life. And then I realized that, well, what was really the most important part of soil was the living aspect of it, the biology. And so I segued 20 years ago into more of the biological aspects of soils. So that's what we're at. We're adding microbiology to that. So you guys, I'm pretty sure you're familiar with the types of biota that are present in soils. We span the gamut from animals down all the way to bacteria. Some of the key players are going to be the guys that are chewing things up, the residue, sort of break down the residues. And these are some smaller organisms that you need to see under a microscope. They're important for litter decomposition and shredding. Then we get down into the smaller organisms, which would be the protozoa. They're under the family sort of protists. I mean, we got a little amoeba and a ciliate and a flagellate protozoa there. Their role in soils is actually to eat bacteria. So they're very important in releasing nutrients that are tied up in microbial cells. Okay? Over here we have one of my favorite groups of organisms. The visual diversity that's found in the fungi is absolutely amazing. And their metabolic or degradative capabilities is very broad. Okay? If we didn't have fungi, we'd be amassed in wood, put it that way. And then on this far part here, these are the real small guys in the soil. We got our bacteria, which can, be up to about 10,000 or plus species. We haven't cultured nearly a fraction of those, but we know they are there from genetic ability to sequence now. And actinomycetes are just they're under the bacterial group, but they have a filamentous, some of them have a filamentous mode of growth similar to fungi. And so functionally, they act a little bit differently in soil systems. So that's just a snapshot of who's in the soil. And I like to break down, if you're going to understand soil biology and how that contributes to soil health and ecosystem services in the more broader context, I like to frame the biology in these questions. First of all, diversity of organisms that are there. That's a key interest of us, or most of us in ecology, because we want to know, does above ground ecology drive or influence below ground ecology? And I just noticed that nice list of diverse cover crop species that were being interseeded. And I'm thinking, okay, over time, is that going to contribute a little bit to more diversity in the soils? Well, the jury's out on that. We'll get maybe back to that later. Biomass is just a measure of how much, so the absolute amount in a weight or a number category. Activity. Now, I heard someone use the word activity, but it was actually in the wrong context, but I'll get to that in a minute. Activity describes totally what they're doing. It's like you're there and you've got a certain amount of mass, and you're breathing. Your mass doesn't really reflect your breathing. Your breathing is going to change if you're running around versus sitting, you know, things like that. So biomass and activity are very different concepts as they apply to all organisms, okay? The next thing I'm very interested in is this location, because we usually, when we talk about soils, we emphasize the physical matrix part, you know, the pore space, the degree of aggregation, we emphasize water infiltration and drainage, those types of things. The microbes are living in that soil, or all the biota, including the microbes. So where they are in the soil is key to what they can do at any one time. Okay? We'll go on, talk about that a little later. Mechanisms just reflects how they're doing a various process, and controls are, hopefully, if we can manage some things. That's my goal is, can we manipulate the microbial community to benefit growing crops or other agendas that we have in the soil. So, getting back to this thing about microbial biomass, it's just an amount, okay, that's the key thing. We usually don't include plant roots, and we skip, you know, your things like your voles and your large fauna. Anything smaller than that we tend to include. It doesn't tell us anything about activity, and in the next slide I'm going to tell you a little bit more about activity. So it's referred to in a lot of textbooks. We talk about it as this black box thing. And really composes three main groups: the bacteria, the fungi, and the micro and the mesofauna. This is some examples of what's in this group, say minus the earthworms, because they're sort of a macro, they're on the macro scale. So we have things like mites, nematodes, protozoa, some algae, fungi, actinomycetes, bacteria. This is going down in size as we go down. And notice what's happening also is when you get down to these groups, you've got a lot in biomass and a lot in number. So the biota forms like an inverted pyramid. You've got your higher level organisms and larger organisms, small numbers at the top, and as those organisms get smaller and smaller and smaller, they can fit in various habitats, then they get more abundant. They also get more diverse in terms of number of species, okay? But overall, if you went and looked in that teaspoon, you're not going to see a microbe in general, because they're really less than 1% of the soil mass. We have a hard time even seeing them under scanning electron microscopes, where we can actually visualize surfaces and go down into pores and look for things, because they're covered in clay and dirty materials, organic matter. So they're not even though we've got lots there, the soil isn't teeming everywhere with bacterial cells. So keep that in mind as we go forward. Okay, diversity. When we talked about diversity before, about the 70s and 80s, we talked about culturing microbes from soil, okay? That was the only way we could measure diversity of organisms in soil, was to get them out and identify them. Well, with the advent of molecular techniques, and this was pioneered by a scientist at Illinois University, where we got these three kind of major domains of life, and we're up in the eukarya. Bacteria are here, and then these are some, they're as old as bacteria, but they're very stress resistant, and we have a lot of those in soils. But when we think of archaea, we often think of, oh gosh, thermal vents, hot springs, really inhospitable saline soils, things like that, okay? So we're knowing a lot more about the organisms that are in soil just by virtue of being able to acquire DNA from those organisms. Okay, this is the one I want to spend a little bit of time on. Microbial activity. I often hear people will talk to me and say, well, my soil has a high microbial biomass, therefore it's very active. Actually, if your soil has a high amount of soil microbial biomass, it means that that biomass has been tucked away and preserved in the soil and is really not very active. It's sort of resting and waiting. So this is why I like to use this metabolic status sort of pie-shaped graph. this is the active portion, and this is going to fluctuate a lot. But it's not going to get much bigger than this. If it did, it would burn up all the soil, all the organic matter in your soil. And frankly, you can't really do that because the microbes would want to grow and they would run out of other nutrients to grow. And third, they'd run out of space to grow, okay? A good example of this is a little bit of a side. I use this for teaching. E. coli, nasty guy, has a bacteria. It's a bacterium. It has a generation time. It doubles every 20 minutes in a lab, okay, every 20 minutes. So they grow exponentially. So 1 makes 2, 2 makes 4, 4 makes 8, 8, 16, blah, blah, blah. Well, if you do the calculation over 24 hours in an unlimited environment, it just took over the globe, okay? So what I'm getting at is most organisms are not dividing. We're not dividing all the time, so why do people think bacteria should be dividing all the time unless we're growing them in a flask and then they kaputs because they run out of space and other things? Okay, so that's the one message, is that in soils you're going to have hot spots of microbial activity, short-lived and highly dependent on resources, temperature, and all these environmental controls, okay? The next thing is, this is a really good example to talk about activity. When we measure activity in soils, what we often do is we want to measure CO2 emission, how much CO2 is coming off the soil. And if you look at this, look at the relationship with soil temperature. So this is a soil that's been incubated, subjected to this increasing temperature, and now you can see how much CO2 is coming off, okay? So you've got the same biomass in this soil. All you've done is change the temperature, and the activity has increased, the respiration rate, but that's not going to last for very long because they're going to run out of available carbon. But in a snapshot, that's the relationship between activity and an environmental variable like temperature. It could be water, it could be something else, okay? So where are they? This is my favorite part because I'm a soil scientist. So I like to think of it in a 3D sort of cartoon here. And then I've listed sort of a bunch of the different macrofauna, microfauna in the soil, and bacteria and fungi. And then I've got their body width here. So we're going from a larger body width, larger over here and smaller over here, okay? Body width is what determines how well an organism can move around and find food in a soil, okay? So this is an example showing a scanning electron micrograph of an actual pore inside a pore, okay? So they've chunked open an aggregate and they've taken some pictures. This is clay mineral, okay? This is a little actinomycete hyphae right over here. This is all polysaccharide stuff that microbes have produced, and there's probably microbial cells buried under there, okay? So accessibility to graze on those microbes in there is determined by how fat you are. Can you get into the pore or not, okay? And most can't get into the pore, So this is really important ecologically on how food webs interact. It's all about spatial relationships and the ability to get access to food. Okay, they're also in aggregates. This is, for some of you, I know we're doing diversity and cover crops here. This is from a continuous corn growing contest winner in Iowa, which is probably not what this is about, but this is gorgeous soil, okay? Just gorgeous. So if we went in here and took this little sample out and then looked at it at a very small scale, like 10 microns, which is the size of 10 bacterial cells. This is what it looks like from the perspective of a microbial cell. So up here, a couple little bacterial cells right on a pore. They like to attach. In here is a closed pore. There's about 10 bacterial cells all squished in there. So these cells, because they're in a larger pore, they would have the opportunity to grow a couple times. So 2 to 4, 4 to 8, 8 to 16. But after 16, I doubt they're going to grow anymore because they can't fit. These guys are stuck. They can't do much more. They got to wait until they get out of there in order to then grow. That's what makes soils so resilient, because we have lots of these habitats where the microbes are tucked away in these pores and they're being protected for generations, okay? They're within decomposing residues. So this is showing a nice example of mite fecal pellets, and they're just teeming with bacteria, and they're decomposing the inner parts of that straw residue. They're within roots. We've got some good examples here of mycorrhizal fungi and rhizobium for nitrogen fixation, but we also have colonies forming on roots at leaky junctions, such as on a root hair. Okay? So those are where microbes like to hang out too. So what are they doing, okay, when they're in the soil? Well, most of them are resting, as we've sort of talked about. But what they're doing really is looking for what we look for. We look for a carbon source. We need our energy, which for us comes from what we're eating, carbon. And then we need this little thing we call reducing power. And reducing power is needed in order to metabolize and build new cells. If you don't have reducing power, you can't make a new cell. And that we get through transformations with ATP. Okay? So what's this controlled by? Abiotic factors, very important, pH, nutrients, salts, all those Haney variables and Ward Lab basic nutrient packages, those are important variables. Soil texture and structure, because that regulates that food web dynamic, the accessibility issue, and then some biotic factors, who likes who and who doesn't like who. That comes into a strong play in the soil. Okay? There's lots of fighting going on. Decomposing plant residues. This is what your cover cropping has a lot to do with this. You've got this living material. It's providing roots to the below ground. It's providing surface cover of the terminated cover crop. But eventually you're going to have it as a residue at some point. Well, a residue, in this case, there's some straw under here. You can't see it. And that's way up at 100% carbon. So we're just tracking this, what happens over time. This is fungal hyphae that you can see, very visual, and there's probably some bacterial cells under there more closer to the straw residue. So what happens when residues decompose is the plant cell carbon is dropping along this line here, okay, pretty rapidly. 60 days, you're almost down to 10% or less. The total carbon, though, in this total mass here, drops much more slowly, because of all that new biomass that grew up on this straw residue. Notice this doesn't level out, okay? It starts to go down. So what that means is that you have microbes and fungi growing on straw, building up biomass in little locations, and then that is being consumed and fed upon new guys coming to lyse those cells. So there's a cycle going on as we degrade residues, okay? And all of this results in CO2 activity coming off our soil, okay? Okay, so what's that? Why is that important? Okay, here's another little graph over here. These are some main components that are found in residues, of plant residues. Cellulose is a big one, hemicellulose, lignin, sugars, amino acids. This is all the simple stuff, protein, fats, and oils. If you monitor over time a residue, what happens is all the easy stuff gets gobbled up first. That makes sense, less energy required, so eat the easy stuff. And this is the hard stuff. This would be my husband saying, oh, do you mean I gotta eat chicken off a bone? I hate that. He wants me to cut it off the bone. So this, yeah, and ribs, let's not talk about ribs. So anyway, those will start to increase over time relative to what is going down with the easily consumable stuff. Okay, CO2 is coming off. Those nutrients like nitrogen, phosphorus, sulfur that were tied up in this material then get bound up in microbial cells. So that's that immobilization phase. We talk about CN ratios and immobilization of nitrogen. That's that part. This is the next phase. As these microbes then say, ooh, I'm running out of stuff here. I better start dying off. They'll start liberating those nutrients, or an amoeba came along and grazed on them, and it's going to excrete out those nutrients. You will liberate your nitrogen, sulfur, and phosphorus, which then can be taken up by the plant. So the microbes are a big gateway for the transfer of nutrients from residues and cover crops all the way into the next generation of new crops growing on that soil. Okay? And they're really good at competing for nutrients. So they're going to get them. Mm-hmm.
How does adding biochar change this whole dynamic?
Let's see, yes. Okay, that could be a multifaceted question. So I think, can I do that after? Okay, but yeah, I'm doing a little work on biochar, and it can do some interesting things. Okay, so let's go on here, because I'm going to get the stink eye from over there. Okay, the other thing they're doing in the soil is, you guys are all aware that greenhouse gases are being produced from our soils. We know that. The producers of those are microbes. So the microbes are responsible for all the methane that's being produced, the nitrous oxide that's being produced, releasing more N2 gas, which is normal. It's about 80% of the atmosphere anyway. So they have a strong role in producing these greenhouse gases, and it's related to aeration. Okay? So this is showing a picture of an aggregate, and I've got oxygen concentration being graphed, 21%, which is like what we're breathing now. And then it goes into 0 as we get into the center of that aggregate. And this is a moist aggregate. So all the anaerobic processes that are producing methane, nitrous oxide, N2 are coming out from here. Over here, some of them are going to get reoxidized again, and that's good, because then they're going to stay in the system. But some are going to escape, nitrous oxide in particular, because microbes don't like to use nitrous oxide. Methane, on the other hand, we're often a sink for methane, because methane being produced is consumed. So sometimes we break even on that one. Okay, this is another fun graph. This is forming soil organic matter, and this is the whole reason I went to study biology in soils rather than sticking to be a soil chemist. So if we think of what happens in any ecosystem, we start with some residue, and then that gets broken down by macrofauna and into smaller pieces. And then there's the activity of the macrofauna as they work into the soil. And we end up with this stuff we call particulate organic matter. Has everybody heard of particulate organic matter? It's kind of that important fraction of carbon that we're interested with respect to management. Then we eventually get to this end, where we're looking at soil humus, this nice dark kind of amorphous stuff. On a scheme of microbial processing, we're going from unprocessed, literally, to some microbial processing, and then lots of microbial processing. This stuff here is largely microbial residues and byproducts of their metabolism. So it's a pretty complex mixture of stuff, and it gets really tightly bound onto clay colloids and things like that, and that's why it's so persistent, and it's an older fraction. This particulate organic matter is kind of fun, because particulate organic matter has some of this in it. You know, microbes are producing stuff when they're living in there, but it still has some character of the fresh residues. So if microbes get access to it, it's a really good food source, but it's not an immediate access. Okay? They don't get it right away. So this is carbon that's more persistent over time, but in the time frame of a cropping season that you can have fluctuations. This, you don't have fluctuations over a cropping season or even 5 cropping seasons. It's hard to do this. Okay? They're also building soil structure. This is a nice little picture of a piece of a particle here, of a residue that's all covered with polysaccharides that are produced by these bacteria. That's helping glue things together. Okay? We want that. This is showing fungal networks, taking smaller aggregates and binding them up to form larger aggregates. Another really important way that we build soil structure. Promoting plant health. This is another area. I don't work directly with the interaction between microbes and plants. I'm more interested in the ecology of the microbes in the soil, but we have a whole plant pathology department that spends a lot of time on this aspect of looking at the interactions between pathogens and beneficial organisms and different plants. This is about as much as I'm going to talk about indicators, is to indicate that everything we measure in soil could be an indicator. Also, there's some indicators from a microbial perspective that are really, really important. And the key one relates to location, location, location. That's the big one. So that's to do with the soil matrix idea that I was talking about, where those microorganisms are. And where they are is going to really determine what they can do. The other ones that are really important are things like pH, salt content, because those are really detrimental at extremes for any organism. So we want to be really cautious about where our pH and electrical conductivity is. The third one would be soil organic matter, okay, because that's going to be part of the food source. And the other ones are all going to relate to water. And that's going to go back to the texture of your soil and the structure of your soil. Can you get water into the soil by good infiltration? Does it move through the soil nicely? Does it then diffuse into the pore space in a more finer textured soil? So are you getting good moisture dynamics going on in the soil system? Those are all going to impact the biological indicators. So how well are we maintaining our biological health by keeping these in check or in optimal ranges? Okay, it's all about the carbon, okay, from a microbe, just like we eat, we want food. So for microbes, it's the carbon. But soil has a lot of carbon, right? You can have 2%, 3%, 4%. You know, go to Canada, you get 6%. Less money, but 6%. That's a bad joke because our dollar's terrible up there. Okay, but carbon has to be bioavailable, okay? It can't just be in a form that they can't use it or attach so strongly to surfaces they can't get it off. Okay, so it's got to be bioavailable. It also has to have conditions. The microbe has to be in conditions of pH, water content, temperature, all those things that are going to make them able to do their job. If they're not, that carbon's going to sit there, the microbe could be sitting right on it, and the two aren't even going to do nothing. Okay? So really what we are concerned about monitoring with respect to managing systems is really the active carbon. And I've kind of put a couple things up here. If you're familiar with how we look at soil organic matter, we have, well, first of all, we have a large pool of what we call humus, and we'll put that in quotes because it's kind of ambiguous, very slowly to turn over. So it's not readily available to the microbial community, although they could burn the entire thing off if many, many years with no other inputs. You'd go from 4% carbon down to almost none over several decades if you let the microbes do that. Didn't give them anything else. Then we have these slower moving pools, and that could be more the POM, the stuff that's protected inside aggregates, and so you have to wait for those aggregates to break open to release that carbon. And the active is more like fresh residues, plant exudates right around the roots, and some of the light fraction that's you can collect maybe by density separation. So these are the things that I've seen used as indicators of biological soil health. Light fraction, particulate organic matter, and differentiating, is it between aggregates? That would be more in the light fraction category, or is it within, so more protected and would have a longer stability in soils. The third, I don't know if you guys have heard of POC. Anyone in here heard of permanganate oxidizable carbon? Okay, this is one that it gets my craw. I was a chemist before I became a microbiologist. And POC is a very mild oxidizing agent. And so there's this trend in the literature, in the scientific literature, to use it as an index of bioavailability of carbon to microbes. Well, microbes don't necessarily like the type of carbon that it oxidizes. Microbes like amino acids, simple sugars, all these sort of smaller molecule things, they don't like aromatics very much. They can use them, but that's not their preferred substrate. POC gets at a more, you could say labile in terms that, yeah, it's oxidizable, but it's not really edible for the microbes. Okay? It takes work and specialized microbes to use it. So if you ever see this become a biological indicator, I would kind of go, well, maybe not. Okay. Let's see, what else? Okay, and then this stuff you guys already know. How do we promote this? I think this is one of the nicest little simplified from NRCS on how to sort of define what we want to do in terms of soil health. But the key things from a micro perspective is we want a habitat. So we want to maintain our good soil structure. We want good air exchange, water exchange. We want protection of microorganisms if we can. If you're dealing with a sandy soil, you've got a whole other root of problems, which is different than we're out here where we're more finer textured soils. We want to limit compaction, obviously, optimize water filtration and holding capacity, provide some type of food that's edible. Okay? So as I said, if you starved your soil and didn't put any, let it go fallow for 20, 30 years, you're going to just slowly be oxidizing all the carbon off in that soil. Okay? It's going to take some time, but you could do it. And then optimize macronutrient and micronutrient availability, things like that. pH, EC, pretty well all the things that are important in soils.